Once again, how are you? How are everyone? This is Mr. Gonzalez, your Saija, taking you through the biolectures. These biolectures are very important to a student who has just joined the advanced level and the students pursuing medical courses. So make sure you listen and then so that you can pick something from there. Don't forget to share and then subscribe so that you can see more of these videos, and also share with a friend, a brother and a sister, and then you see how best you can save the Ugandan. Our mission is saving millions of scientists to excel. We want millions of scientists to excel in this field here. So not wasting time, I'm going to just begin the biolectures. And as I told you earlier, this is Mr. Alex Osaija, taking you through this. Now, what we're going to begin with, we have what to call, we're going to look at what to call the structure and the function of what to call the mitochondria. The structure and the function of the mitochondria. This is how we're going to begin it here. We are seeing all of this structure we are seeing here. The whole of this is the cell. It can be a cell from an organism, but majorly we're focusing on living organisms, and it's what we call the human cell. You get? And therefore, this cell here has been extracted from any part of the body, any part of the tissue. Could be the muscle cell, could be the hair cell, could be the heart cell. Any cell is what we're focusing on today. Now, we are aware that this is the cell here, and of course, the cell is having these structures we are seeing here. These are what we call now the cytoplasm. The cytoplasm. The liquid we see here is what we call the what? Cytoplasm. And these droplets we are seeing here are what we call the what? Cytoplasm. Now, this cell here is often what we call the nucleus. So what we call the what? The nucleus, the one we are seeing here, is what we call the what? The nucleus, with its DNA and then also the nucleus that is inside the nucleus there. We have now the center, what we call now the power of the cell. The power of the cell is what we call the mitochondria. This is called the what? The mitochondria. Allow me to label it here. I'm going to label it as mitochondria. This is what we call the mitochondria. Mitochondria. Now, my brother who's there listening, this is the powerhouse of the cell. Why do we call it the powerhouse of the cell? This is because this mitochondria here, it is very important in participating in the biochemical reactions that involve the yielding of what we call the ATP, of which this ATP here is the energy, is the power of the cell. The cell cannot run its metabolic reactions minus what we call the what? The ATP, which is coming from what we call the mitochondria. That means the mitochondria is a powerhouse because it does what we call several reactions that involves what we call the synthesis of what we call ATP, which is the food or the source, which is the power, which is the energy of the cell. Just like you can be having a phone cannot work minus the battery. If the battery runs away or goes off, that means that you can no longer have the phone working. Surprise the radio, the batteries. Let's continue and we see. This mitochondria here, members, it is having what we call a covering. What's the name of this covering? This covering here that you are seeing here, this enclosing covering here you are seeing here, and this one here, is what we call this one we are seeing here, is what we call the mitochondrial envelope. Is what we call the mitochondrial envelope. Let me put it here. Mitochondrial envelope. Mitochondrial envelope. Envelope, something like this. So what we call the mitochondrial envelope. This covering here. But this covering here is consisting of two layers. It has two major layers that are enclosing the inner part of the mitochondria. This is what we call the outer layer. This is what we call the outer mitochondrial layer, which is this one here. This is the outer mitochondrial layer. I'm going to abbreviate it like this. Outer mitochondrial layer. You get? Or envelope. Now this outer mitochondrial envelope is having something interesting. I'm going to look at it because it is very important in performing very functions. But what you have to know about this mitochondria, outer mitochondrial layer, It is having what to call both the proteins and also what to call the proteins and also what to call the phospholipid bilayer. It is having what to call the phospholipids that are double layered. So has what to call the phospholipids, and also what to call the proteins. Now when I talk about the proteins, I'm meaning several structures. This could be enzymes, this could be carriers, this could be receptors, you get? But all of them are proteins here because you know that enzymes are proteins in nature. Okay, most enzymes are proteins in nature because there are some enzymes which are made up of other structures like the lipids and so on, or the steroids. Now, but mostly the enzymes are, those ones are hormones, sorry. Enzymes are majorly proteins in nature. It is only the hormones that are in different formats that we have, the ones that are carbohydrates, the proteins, and also what to call the ones that are in the lipid form. But for the enzymes, they are majorly proteins in what? In nature. Sorry about that. So, but also we have to know that the ratio of proteins to what we call phospholipids to proteins is the same as 1 to what? To 1. What does it mean here? It means that the proteins in the on the covering here, on the outer membrane, and the phospholipids are in the same composition. We shall also see that this outer layer here is consisting of what we call the proteins that are on the outer layer here. They are major transporters and then receptors, and then we normally just call them what we call the toms and what we call the toms and then the sims. The toms, they are called the toms. These are called what we call the toms, and also what we call the tims. The toms, this is just what we call the translocase of the outer membrane, complex structures. These are what we call the translates of the outer membrane, complex structures. These ones are given in different ways. They are different components. These ones are like around 7 of them. We have what we call the 2 that normally participate in signaling. They work like receptors. For example, the tom 40 and then the tom 70. These ones are work what we call the receptors. All of them are located in what we call the outer membrane here. Then when we begin from what we call the tom that is 5, the tom 6, the tom 7, the tom 22, and also 1, 2, 3, 4. Something like also the tom 44. This one is measured what we call their transporters. They translocate the materials passed through them. They are complex structures. We shall not go into details to see how they work, but just know that this outer membrane of the mitochondria is consisting of what we call the phospholipids. It is having what we call the proteins, the integral proteins. We have what we call, which are called the toms, those that are outside. Then the ones that are on the inner membrane, they want to call the tims here, the translocase of the inner membranal complex or structures. Now, we should also have to remember that this outer membrane of the mitochondria is also fully permeable. It is permeable. That's what we call permeability. Permeability. This is the structure. This is what we call permeability. So we say it is fully permeable, or it is highly permeable. It is highly permeable. Another one is this one is highly permeable because it is consisting of these toms here. These toms here, these structures here, which is that these toms are like proteins in nature. Now these toms here are like in 2 formats. We have what to call the beta and then what to call the alpha. The beta barrier structures of the outer membrane, what to call the beta. Now this beta here, they always have what to call some the diameter is a bit long, the channels are a bit wide. You get the channels of the proteins that are on the outer membrane are what? Wide. That means large materials can easily pass through the outer membrane. You get? And also they have what to call some tiny structures. When I talk about there's something, when I talk about the beta, we have what to call the beta barrier, we have to call the beta proteins. We have to call the alpha proteins. This one is here as secondary structure of the proteins. The beta proteins, this one here, on outer membrane are so complex. They have bigger channels. But the alpha, they have what called smaller channels. And then they allow smaller molecules to pass through them. That means the outer membrane is consisting in major of the beta proteins. You get? Now let's continue. We have seen that outer membrane, once again, is fully permeable, or it is highly permeable. Then it is consisting of what we call the proteins, integral proteins, which are normally called the toms. The toms, we have what we call the beta and what we call the what? The alpha proteins. The beta proteins, this one is have what we call wider channels for passing the materials. Another one is large materials normally pass what we call the outer membrane because of what we call the beta proteins. And then the teams, this one is majorly on the inner membrane. That is it now. Then when we go to what we call now, this one what we call the intermembranous space, what we call the intermembranous space. Intermembranous space. This is the space which is located in between the inner membrane, what we call the outer membrane. So this one is just majorly consisting of what we call, it has what we call the protons. It is the center of what we call inclusive levels, what we call the protons. It has a lot of protons that are inside there that brings about, it is this that normally bring what we call the electron transport system, as I'm going to see later. Then the inner membrane is just invaginated into what we call the structures which are protruding, invaginating. We say this is what we call the cristae. What we call the cristae. This is what we call the cristae. We can see here. We have this. We have this. All these ones are what we call the what? The cristae. Now, we don't have to know that this cristae here, we are going to see that they are very, very important during what we call the electron transport system. Is why they are going to participate a lot. Because the cristae here, when we examine and look at this one here, we have this one that we are seeing here. This cristae we are seeing here. Hope you are seeing that. This cristae is having what we call, this is what we call the oxysome. You get? This bulbous end, in other words, this neck of the cristae is having what we call this structure here, which is called now oxysome. I'm going to label it here, oxysome. Now this oxysome here, on its location here, there is something what we call an enzyme called the ATPase synthase. ATP synthase enzyme. Synthase enzyme is located where? On the bulbous end of the cristae. On which part? On the oxysome. Now, also on this, they want to call some complex structures on the cristae. These are complex structures that are very important in electron transport during what we call the ETS. That means we shall see that this one here, the cristae, has what we call the complex structures, CS, complex structures, which are going to be important, what we call the what? The ATS. You get the point. And then also have what to call, of course, inside here, we have seen what to call the outer covering. Is what to call the outer membrane, which is what to call the outer membrane, mitochondrial membrane, and also what to call the inner mitochondrial membrane. Then what to call inter mitochondrial space. Then we have what to call the cristae, which are an invagination of the inner membrane. The inner membrane is members, is partially permeable. It is partially permeable. Inner membrane, this one here. Where can I extract it? The inner intermembranous space. Once we have to call the inner membrane, this one here. The inner mitochondrial membrane. This inner mitochondrial membrane here, one, it is partially permeable. It is partially permeable. Then secondly, it is consisting of what we call certain protein carriers and transporters. These are number what we call the teams. The translocase is what we call translocase of internal or of the inner, translocase of the inner mitochondrial membrane. These are proteins, these are structures. They are divided into two categories, structures you see here. The translocates, these are like enzymes. Others work like enzymes, others work like protein carriers, others work like binding sites. You get? Now, we have seen this one here. And of course, we have seen that it is consisting of this crystal here, and then also the oxysome, then what called the complex structures, then what called ATS takes place within this complex structure here. Now, when we continue inside, we shall see that there is something interesting there, which is what we call now the matrix. The liquid is what we call the what? The matrix. The liquid is what we call the what? The matrix. This matrix here is consisting of what we call certain suspended particles, where we have what we call now, so what we call now the DNA. So what we call the DNA. The DNA is here. I'm going to put it somewhere here. DNA. Before I go here, there's something I've forgotten here that on the outer membrane here, these are some of the proteins you are seeing here. These are what we call the polys. These are what we call the polys. These proteins here, they are what we call the what? The polys. Now, getting back to the inside, how what we call the DNA, then you have what we call the ribosomes. These are what we call the ribosomes, because this mitochondria here is good at manufacturing its own proteins, though it can also import something what we call the what? The ribosomes, or the ribosome if this one. These are the 70 subunit ribosomes that we have inside here. Then of course there are very many structures. Some lipids also there, and very many others. You get? Now I want us to look at now the functions, because our point is the functions and then the structure and the function of the mitochondria. I want us to begin looking at the functions of this mitochondria. Don't forget you're looking at the mitochondria. We are saying the mitochondria is the powerhouse of the cell. Why? Because it does several metabolic reactions, biochemical reactions, which result into what we call the synthesis of what we call ATP. This ATP is the driver of the cell. So by providing the driver to the cell, we say is the energy power of the cell. You get the point? Let's continue. Then we say that the mitochondria has two major layers. Has the inner and then the outer layer. Outer layer is what we call the outer mitochondrial layer, which is fully permeable, has proteins, and also which are called the tomes, has what we call the phospholipids, and also there. Then we say that the inner membrane is majorly consisting of what we call the teams, and also an invagination or invaginations, which are called the cristae, where we have what we call the certain enzyme called ATP synthase, and also what we call the oxysomes, which are very important to call what? In the electron transport system. Now, we are going to look at, now we have seen the parts that are inside the mitochondria, like we have seen what we call the matrix is having what we call, is a liquid, where most of the chemical reactions like glycosyl takes place inside here, the matrix. Then we are seeing that this one, crystal is for the ETS, electron transport system, DNA is for the precursor, and then ribosomes also for protein synthesis and other enzyme synthesis there. So we're going to begin by looking at something we normally call now the functions of what we call the mitochondria. Now the first function is supposed to be going to be what we call the apoptosis. The apoptosis. And this apoptosis here, it is the same as what we call programmed cell death. Remember, it is very important for the cell to destroy what is not important, so as it can allow the growth of other what? Structures and other cells. This is what happens here. We know that most of this is what we call now, this is the cell that we are having here. This is the cell here that I've extracted it from here. This one is this cell here, the whole of this one here, but I've just made it small. Now this cell here is containing what we call the mitochondria, the energy power, or the powerhouse of the cell. The nucleus is here. You get? But what's going to happen here? There is something interesting. Now, we know that during what we call apoptosis, we have certain structures here, like what we call the cytochromes. cytochromes, cytochrome C. Now, we have what to call certain enzymes that are important in what to call destroying the cell. Now, what happens is that during this apoptosis here, that the cytochrome, and this is because it is in two ways. We have what to intrinsic and what to call the external or extrinsic pathway, or the mechanism taken to destroy the cell. But this one I have a video about it. I have a video that is talking about the exact process, but I'm just going to summarize it just briefly. Now, what happens is that when the cell detects that there is any structure inside it that is not normal, or the cell is no longer functioning, what's going to happen here? This cytochrome here is going to leave the intermembranous space or the outer membrane of the mitochondria, and it's going to move inside this side here. Reaching inside here, it is going to activate. It's going to activate certain. It's going to burn. These are how to call certain. It's going to activate what to call. It's going to activate certain enzymes there. This is the enzyme which is going to be activated. So after activating this enzyme here, it is going to move towards what? Towards the cell membrane here. Also, towards the mitochondria, through these pores of the mitochondria space. So this enzyme is going to move like this. Then reaching there, it is going to begin destroying what we call the worn out tissues of the cell. It's going to begin destroying the cell. You get? Most especially when the cell is what? Is mature. I have a full video of this one here. We're just passing through is what you have to know is that the apoptosis is what we call the programmed what? Cell death. So let's go to the next part now. We have what to call now the next function is what we call the protein transport. The transporting of the proteins. And which part of the mitochondria is very important in transporting what we call the proteins. Let's see here. Now this one we're having here is what we call the mitochondria. This one here is the nucleus. This one is the cell. You get? Now this one is what we call the inner, the matrix, mitochondrial matrix. Mitochondrial matrix. Then this one to call now what we call the outer membrane. That's what this what we call the outer part. What we call external membrane. Or we can say this is the same as what we call the cytoplasm of the cell. We are aware that, remember, there's something that takes place here. In the cell here, in the nucleus here, there's something that normally takes place. What takes place here? We shall have what to call, in the nucleus, we have what to call transcription takes place in what we call the nucleus. Transcription, whereby the DNA is converted into what we call the RNA, which is this one here, the RNA, the messenger RNA. This one is going to leave the nucleus and goes to the cytoplasm. Reaching the cytoplasm here, we have some organelles present here, and this organelle is what we call the ribosome. This is what we call the ribosome. So this is going to bind with this ribosome here, and then to manufacture what we call certain proteins. But these are inactive proteins. These are what we call inactive proteins. These are what we call inactive. They are inactive proteins. So what is going to happen is that these inactive proteins here, they are going to be moved into the mitochondria to be activated. You get? So what is going to happen is that these inactive proteins here, they are going to move and then they pass through what we call certain channels. Before passing there, they reach on what we call these receptors. You get? So these are called the TOMs. The translocates of the outer membrane. Remember I said they are of two types. We have what we call the T. We have what we call the T20 then also the Tom 20, and also what to call the Tom what? The Tom 70. So these ones here, they're on the membrane here. Their major function is to detect or to to receive, you get, the precursors. The precursor is that inactive protein, inactive structure, inactive enzyme. So that means this one here is an example of what to call what? The TOMs, the T20 or the T70. Now after detecting that this one, what is coming, is a precursor, then what are they going to do? They are going to open these channels here. And then after opening, this one is going to pass. It is going to pass through this and it goes into the intermembranous space of what called the mitochondria. Then reaching there, they are going to again be detected, the proteins. They are inactive. They are going to be the what to call the TIM, the translocase of the inner membranous complex. So this TIM here is also of two types. We have what to call the T35, so what to call the T37. So what is going to happen is that we shall have what to call, after binding it, this is going to open these channels. This channel is going to open and then we shall have to call the proteins to go into this. Now this protein is what is going to happen here. They are going to find certain enzymes that are still sitting in the mitochondria. So these enzymes here, they are going to activate them. So we shall have, they are going to come and then bind onto these enzymes here. So after binding on these enzymes here, this enzyme here, so we have what to call this and this. So this is going to activate them. So we have what to call now the fully activated protein. This can be molded into an enzyme to be used by the body, or it can be remain as a protein to be used by the mitochondria, because the mitochondria has a tendency of manufacturing what we call the proteins. But now, it means that most of this protein, this one hasn't been imported. It has come from the nucleus. But you also have that this mitochondria here is also very funny because it has the DNA on its own, has the DNA. So this mitochondria, because have what called the DNA, what's going to happen here? It is going to undergo what called transcription from what called the messenger RNA, which is going to bind with what called certain ribosomes here. Certain ribosomes that are going to be bind with this ribosomes. So after binding with this, they form what to call translation. Then they form what to call the what? Also proteins here. But the proteins that are manufactured generally inside the mitochondria by the DNA of the mitochondria, they contribute almost like 15% of the total proteins that are used. That means the remaining 85% of the proteins, these are just imported. It just manufactured what we call the synthesized one. That means the mitochondria just manufactures 15%, but the remaining 85% just comes from what? From the outside the mitochondria. That is it now. This is what we have so far seen that the mitochondria is important in programmed cell destruction, also called apoptosis. They say that mitochondria is also important in the protein transport. We also have what called the miscellaneous transport, also called the metabolite reactions or transport. Now we are aware that members, this is what we call the membrane of the mitochondria. This is the outer mitochondrial membrane. This is what we call the inner. This is the outer or this is the inner mitochondrial membrane. What's going to happen here? Something very interesting. Now, we are aware that outside this is the cell, and this is what we call now the cytoplasm of the cell. Hope you're together there. This is what we call the matrix, the mitochondrial matrix. What is going to happen is something very interesting. We know that during glycolysis, we form what we call the pyruvate. The pyruvate is formed in what we call glycolysis. This pyruvate here before it goes into the mitochondria here, it is first decarboxylated. You get? Decarboxylated to form what we call acetyl coenzyme A. That means that what is going to happen here? Now what's going to happen is that I'm going to have my structure here. Now, I have my structure here. This is what we call the outer membrane, this is what we call the inner membrane. And what is going to happen is that these are just channels, protein channels that we are seeing there. What happens is that this mitochondria here, it allows the influx or the flow of what we call certain chemicals. What is this one here? What we call the acetyl coenzyme A. What's going to happen is that means that the mitochondria has participated in something that we normally call the conversion of what we call the pyruvate into what we call the what? Decarboxylation it into what we call acetyl coenzyme what? Coenzyme A. That means this is now the first biochemical reaction. That is conversion of what we call the pyruvate. That means this is number one. The pyruvate is being converted into what we call what? Into the acetyl CoA. This is a biochemical reaction. But remember, this acetyl coenzyme A and even some other products there, they can again go back and again form what we call the glucose. This is what we call now gluconeogenesis. That means the second arrow that is moving down on this side here is what we call gluconeogenesis. This one here, gluconeogenesis. The conversion of the non-carbohydrates into carbohydrates is what we call gluconeogenesis. Now, we have what we call, remember, the amino acid that also normally fall inside here. The amino acids. But these amino acids here, they have what we call amino group, which is so toxic. So this amino group here is going to be undergo what we call the urea cycle. The urea cycle. In other words, removing the converting the amino group into what we call urea, the less toxic, because urea is less toxic, but ammonia is more toxic. So the process of converting, removing the amino group and then converting it to what we call the less toxic, this is what we call the urea cycle. That means that also here we shall have what we call the urea cycle that is going to take place inside the mitochondria here. That means also have what we call number 3, what we call the urea cycle. But also, so that there was a after forming this acetyl coenzyme A, it undergoes what we call several reactions, forming different samples. We don't want to talk about them here, up to oxaloacetate. Now, what's taking place here is what we call the Krebs cycle, also called the tricarboxylic cycle. That means that the mitochondria, starting with the acetyl coenzyme A, it has undergone several metabolic reactions to give out several substrates, and therefore that means that it has also important what we call the Krebs cycle. So the first supposed to be the what? Krebs cycle, the tricarboxylic what? Cycle. We also have that there is a tendency of these fat acids flowing into what we call the what? Into the mitochondria. There are channels for the flow of the fat acids into the mitochondria. But this fat acid, what is going to happen is that they are going to be decarboxylated. In other words, they are going to be, these fat acids are going to be converted into what we call acetyl coenzyme A. By which process? So as you can get energy from it, also as can be converted back into glucose, or it can be another what we call the Krebs cycle to get what we call some more ATP or energy. This is what we call the beta oxidation. Beta oxidation process. The process of converting fat acids is what we call acetyl coenzyme A. That means also what we call beta oxidation. Then, of course, also have what to call some of these acetyl coenzyme A can be used to make what we call some ketones here. You can also convert them what we call the ketones. That means I say ketogenesis is very important. Ketogenesis. You can also make some what we call some heme. When that's normally found in the blood. That means we call heme synthesis. So what you have to know that most of these reactions here, they take place in both cytoplasm and also both the what? Mitochondria. You have to note that. Because some reactions do not complete their cycle. Like the urea cycle does not complete its processes in the what? Mitochondria only. Some processes have to take place in the what? Cytoplasm. You get? So you have to note that. Now, this is now that means that we have seen what we call now the metabolites, transport, what we call the machinery transport, transporting the end products. Another one is the substrates, and also some other products. So let's see what we call now another important is, which is called now the ATS, electron transport system. That one is how does the mitochondria participate in what we call the ATS. There is something you are seeing here. This one you are seeing here is what we call now. This is this structure here. This one is what we call the inner membrane. This is what we call the inner membrane. Inner mitochondrial membrane. Now, this membrane here is having what we call the inside part of it, which is what we call now the mitochondrial matrix. Mitochondrial matrix is given as what? MOM. This one is given out to call the inner membranous space. Inner membranous space. Or inner mitochondrial space. Just like what we are seeing here. This one, all of this one here is the same as the one we are seeing here. The inner membrane, this one here. Then this one is the same as this one here. Then this one is the same as this space here, which what we call the what? Membranous space. Don't confuse it. Now what's going to happen? Remember that we know that during Krebs cycle, the pyruvate is converted into what we call the acetyl coenzyme A style CoA, and then undergoes what we call several reactions, what we call different substrates. Then during this process here, we have what we call the formation of what we call the energy carriers, like so what we call the NAD H2, and also what we call the FAD-H. What's going to happen? These are carriers. Now, what happens that this is what we call the Krebs cycle. Now, when this NAD H here, remember here, we have what we call a lot of protons here. We have a lot of protons in the mitochondrial what? Matrix. Very many of them. Very many of them. What is going to happen is that this NAD H is going to become oxidized. It's going to release what we call the protons and what we call the electrons. It's going to dissociate and give what we call the electrons and also what we call the what? The protons. What's going to happen here? This NADH2 is going to give what we call the electrons. Now what's going to happen that these ones are going to remain, of course, are going to remain inside here to increase on the concentration of what we call the what? Hydrogen ions. Now, this one is a complex. These are complex molecules, which look at them when they come to ATS in details. What happens is that these electrons here, after being formed, they are going to immediately gain some energy, and then they are going to move onto this complex here. This is complex 1, the energy complex 1. This is going to become excited, what we call excited. After becoming excited, what's going to happen here? There's something very interesting. It is going to open the channels, and then it's going to allow the proteins from the mitochondrial matrix, this one is here, to flow, and then they concentrate around here. So after finishing that, what's going to happen? And then this chain is going to continue like that. Then we shall also see it. This one is going to again, we have what to call certainly electron carriers here. The coenzyme Q, what to call cytochrome C. What's going to happen is that this one is exciting this one here, it's going to become excited, and you shall see energy is going to go to this. This is going to excite this one here, and then this one here, and then we shall see that now a protein is going to be accepted by this, by oxygen, and then oxygen is going to break down into certain atoms, then which is going to form what to call hydrogen, form what to call what? Form what to call water molecules, and then we shall see that the last electron acceptor is what to call the what? The oxygen. So the oxygen is going to, remember this process takes place in the process of what to call oxygen. So what happens is that the electrons from this complex here was accepted by oxygen. You get? And therefore the oxygen atoms got reduced, and then they broke down, and then they combine what called hydrogen atoms to form what called this water here. So then what's going to take place here? We have what to call this water, which is what to call what? This one here we are seeing here. This get here, this structure here, remember here, whenever this one gets excited, what is about to call the protons are pumped to this side here. By the time we reach, that means that we have a lot of hydrogen ions here, the protons. But this side we have what to call less protons, which are there. So the difference in the concentration is going to force what to call the protons to move through what to call this crest here. Because they can no longer pass, these channels are closing. The membrane is closed. That means they can only use this channel here, which are the bulbous end that we saw here. This channel we are seeing here, this part here, is the same as this one here. So they are going to begin flowing. As they are flowing, what's going to happen? We have what to call the ADP that is waiting to come out to call what? Organic phosphate, to give out to call ATP. But the challenge is there is no any energy that is going to activate this enzyme here. The ATP stays. It is there, but it's lacking the energy. So as these electrons, as these protons are flowing along this side, they emit what to call some energy. That energy is going to help this ATP synthase enzyme to bind or to combine the ADP with what to call inorganic phosphate. The adenosine diphosphate to combine what to call the phosphate form what to call what? Adenosine triphosphate. Now, we have seen that this ATP has been formed by the process of what to call oxidation. There has been what to call oxidation and what to call reduction. This FAD H, it only works major on what to call the second what? On the second complex there. By like the way this one, like the way the NAD works, is the same way, emitting the hydrogen ions and also the electrons which go here, and also go and combine the one that is here, because the ones from this are going to go directly into this one here. So the ones from the FAD are going to go into this and then excite this, but this one here does not share to the next. It's just going to give this one here, the coenzyme Q, which then is going to go and carry those electrons and go and excite this one here, the complex, third complex, from this to this, then from this to this, and then we'll see what's going to happen next. So the process by which we have formed ATP here through a series of oxidation, whereby we oxidized, we got oxygen, and then we oxidized some of these complexes there. This is what we call oxidative phosphorylation. Now, the last importance, that means the last importance of what we call the mitochondria is what we call the replication and transcription. It is something very simple. I also look at it here. Now, replication, know that at the first is some theory which is called endosymbiotic theory, whereby it is said that this mitochondria was a prokaryotic organism, but was invaginated by a given eukaryotic organism. Then it changed into an individual organism. So this mitochondria has what's called the DNA. This DNA here has a tendency of undergoing what we call transcription to form what we call messenger what? RNA. This messenger RNA here, what is it going to do? It is going to undergo what to combine what we call the ribosomes, and then it's going to what to call what? What we call translation. Translation to form what we call now the proteins. You get? Now, this is what we call transcription, the conversion of the DNA to messenger RNA or the RNA. What we call transcription. This is what we call transcription here. Transcription. This is what we call now converting this to give out called proteins here. This is called now translation. Then we know that this mitochondria here, it has a tendency of giving, replicating to form what we call individual mitochondria, a process which is called what? Physiology, just like the one in the bacteria. So in brief, we have seen that the mitochondria is the energy power of the cell, and then I said that this mitochondria here, it is having two major structures: the outer cover and what we call the inner membrane. Then the outer membrane is having what we call the membrane and also what we call the matrix inside. Generally, that's what we call summary. It's called the summary. So the mitochondria is consisting of what we call the envelope, the mitochondrial envelope. Then it plus also what we call the what? Plus what we call the matrix. So this envelope is consisting of what we call the outer, also what we call the what? The inner. The inner is having what we call the what? The crystal. Then the matrix is consisting of what we call the ribosomes, the DNA, and others. Then we have seen that now the functions of the mitochondria. The functions of the mitochondria. We said one, it is for transporting of what? Transporting proteins. Protein is what? Transport. We say protein transport. We have seen that. Then next say that this also for miscellaneous transport, means miscellaneous transport. Then we also say that this is so important in ETS. ETS. Then mitochondria is also very important, what to call the replication and transcription. Replication and transcription. Then those are what to call the features, the functions of what to call the mitochondria. Thank you for watching my videos. Please make sure to subscribe, like and share so that you can see any of the videos. You're free to write in the comment section to ask any problem that you want me to do for you. Once again, this is Mr. Gos. Thank you for listening. Bye-bye.